Good evening. Let's start today's session. Let me share my screen. So last week, this Tuesday only, we studied about hydrophobic interaction chromatography and ion exchange chromatography. So this week's content will mainly have uh, size exclusion chromatography and affinity chromatography. Okay. So just to brush up, based on if we want to explore the charged, like we want to separate or purify our molecule of interest or protein of interest based on the charge, then ion exchange chromatography or chromatofocusing can be done. Chromatofocusing exploits the isoelectric point or pi values of proteins. Then based on the charge or hydrodynamic radii, one can use gel filtration or size exclusion chromatography. Hydrophobicity, we looked at hydrophobic interaction chromatography. And today's session will cover affinity chromatography as well. Now, size exclusion chromatography mainly separates different molecules of various sizes as they pass through the SSC matrix or resin, which is packed in a column. But unlike ion exchange or affinity chromatography, in case of size exclusion chromatography, the molecules do not bind to the resin. Okay, do not bind to the matrix, and buffer composition also do not directly affect the resolution. We looked at resolution in great detail, which is basically or shortly, briefly, the degree of separation between two peaks. Okay. So what is the advantage of SEC? Like size exclusion chromatography is also like abbreviated as SEC. The significant benefit or advantage is that the conditions can be varied to suit the type of sample or molecule of interest or the type of protein we wish to purify, the user wants to purify, or the requirements for further purification. If you want to really analyze um, like macromolecular complexes, their affinity, their binding assays, all these can uh, include SEC as one of the steps. Okay. So it is actually a highly versatile separation technique, and it is actually very much suited for biomolecules which are really sensitive to changes in pH, concentration of ions, mainly divalent ions or monovalent ions in general, or any cofactors or harsh environmental conditions. It can also be separated in the presence of essential ions, cofactors, detergents, urea, or guanidine hydrochloride at high or low ionic strength. SEC can also be performed across a wide temperature range. Many times, in general, the columns that we were talking about, all these chromatographic columns, can be reused if it is maintained in a proper condition. Okay? So after every use, one should, what we looked at earlier was ion exchange chromatography and hydrophobic interaction chromatography, right? So for those columns or even for SEC columns, what actually is done is once one prep is done, one protein purification is done, it's always a good idea to wash it completely, then pass high salt and like NaOH, like strong alkali, through the column so that anything what is bound to the matrix is stripped off. And then we wash it with water again so that the bound NaOH or remaining NaOH or salt is washed off. And then we store the matrix in 20% ethanol. Okay, this is the ideal way to store a chromatographic column. Okay. And then once again, when you want to use it for another protein prep or for another, for any other protein or nucleic acid or anything, what you can do is you start again with washing with water and then um, equilibrating the column, equilibrating the resin, and then loading your sample. Okay. So all these matrices or resins are actually not only SEC in terms of other matrices as well. Usually, storage at 4 degree keeps it intact for a longer time. But many times SEC is also SEC columns, you can see that people perform SEC at room temperature as well. Okay, in between 25 to 30 degrees Celsius. And even there are some columns, SEC columns, which enable separations at even higher temperatures. Okay, and the purified biomolecules that we wish to purify, the molecules of our interest, can be collected in any chosen buffer. Now, when we talk about SEC resins, so the porous matrix is chemically and physically stable, and it is actually packed with spherical particles having properties that minimize the adsorption of biomolecules. We talked about it previously that in an SEC resin, the molecules that we wish, that sample of our interest, it doesn't really interact with the matrix. All the molecules or the samples that all the molecules in our sample that we load on the matrix or on the column do not interact with the matrix. It is mainly separated out based on the size or the overall hydrodynamic radius. And the packed bed means the matrix, the bed volume, is equilibrated with the buffer, and the same buffer is actually used throughout. This buffer, the equilibration buffer or the running buffer, fills the matrix pores or resin pores and the space between the particles. And the liquid which is inside the pores or the stationary phase is in equilibrium with the liquid or the mobile phase outside the particles, right? And sample components actually are eluted isochronically, which means the buffer composition remains constant throughout. In the earlier, even with affinity chromatography that we will look at later, as well as the previous ones that we discussed in the last tutorial, the hydrophobic hydrophobic interaction chromatography and ion exchange chromatography, all these have different buffer compositions, right? Initially, at low ionic strength, the buffers are prepared, and then while eluting the sample of our interest, while eluting the protein of our interest, the ionic strength is increased, right? In case of ion exchange chromatography. In case of affinity chromatography also, the elution buffer and the equilibration buffers are different, okay? But in case of SEC, this is not the case. That's why it's called an isocratic elution, where the buffer composition actually remains same. It is the same buffer that is used for equilibration. It is the same buffer that is used for elution as well, okay? So buffer composition remains constant throughout the separation, and there is no need to use different buffers during separation while doing SEC. And a wash step is used using the same buffer, the equilibration buffer or the running buffer, at the end of the separation, which actually removes some molecules that might have somehow retained or might have been stuck in the matrix while coming down, or I mean coming out. It didn't come out properly. So if we run excess of the equilibration buffer or the running buffer, eventually any remnant molecules will elute out, okay? And then it has to be washed properly. Not only what I was talking about, not only this wash step using the running buffer should be done after your sample of interest has eluted out, so that any remnant molecules come off, and then proper washing should be done, followed by storing the matrix always in ethanol, okay? It also helps removing, avoiding the growth of any kind of microbes. And actually, this gel filtration or size exclusion chromatography can be performed directly after any other chromatography step, like ion exchange, hydrophobic interaction chromatography, or affinity chromatography, because the buffer composition usually does not affect the final separation. Okay, the separation is based on the size of the constituent molecules. So, if this is the matrix, this is the mixture of molecules that we wish to separate. So what is happening here is, you see, there are three different kinds of, three different sizes of particles inside this whole mixture, right? The red ones, the yellow ones, which are the smallest, and this orangish ones, which are somewhere in the middle, like somewhere having intermediate size. Now, if this gray one, these are the subsequent steps of SEC, what is happening here after they load the sample. So your complete mixture is here. This particular band that you see here, this gray colored strip is the matrix SEC resin, and this complete mixture is loaded. Okay, this is having all these three types of molecules here. Now, as the mobile phase keeps on passing through the column, SEC column or SEC resin, what happens here is the resins are packed in such a way that through the pores, uh, because the pores are regulated in such a way, the smallest molecules can pass through the resins, can pass through the pores easily, like it can get inside the pores. However, the bigger molecules will have a tough time getting inside these pores, and hence what it does is it does not really get inside the pores, it directly passes outside the pores. Okay, comes out of outside the pores. So the smallest molecules which are going, like which will eventually, due to their smaller sizes, can eventually get access inside the pores, and hence it will take longer time to elute as compared to the larger ones, like larger size molecules. Okay. So this is what is happening here. The largest ones, the red ones here, um, these are um, like passing through the matrix more with much higher speed, like it is passing through quickly as compared to these yellow ones, the smallest ones, which are actually having the access to all the pores of the matrix, and hence it is taking a longer time to go through the resin. Okay. Eventually, uh, these red ones actually will elute out earlier than the other types of molecules. Okay, so this is the point. Like this is the absorbance. We talked about, suppose it when we are talking about a protein. So the sequence of amino acid dictates the protein structure, right? So the amino acids, the aromatic amino acids, tryptophan, phenylalanine, and um, tyrosine, these three actually show higher UV absorbance, right? So what happens here is, this is what is measured, like higher the amount of a higher content of the protein, higher content of that particular amino acid in the protein chain will lead to um, these peaks here. Okay. So first you are injecting your sample, having this mixture. This is the same elution profile for this. Okay. Now what is happening here is this is a high molecular weight. The red one is passing through this. Okay. And then the intermediate ones, the orange ones, comes next, as you see here in this strip, the resin or matrix. And then the lowest molecular weights will take the highest time or the longest time to really come out and elute out of the SEC resin. Okay. Now, when we talk about preparative size explosion chromatography, high resolution size based separation of biomolecules with fractionation, preparative SEC actually isolates one or more components of a sample. It separates the components which can be directly transferred to a suitable buffer for assay, or it can eventually be stored as well. Okay. And small sample volumes actually for SEC, usually even if you purify a huge amount of protein for other columns, right? For SEC, usually you can load up to like 0.5% to 4% of the total column volume. Like how much is the packed matrix or packed resin, volume of the packed resin in the like SEC resin? 0.5% to 4% of the total column volume can be applied, and at low flow rates while using long columns. Okay. And these are just few information, practical information, which is to be kept in mind. All these points are to be kept in mind when you are actually performing SSC in the laboratory. Okay. And particle size commonly greater than 12 micrometers, providing low column back pressure, and the resolution, basically the separation between the two peaks during preparative SEC varies from high to more moderate, depending on what is mainly the size of the particles constituting your sample. And for optimum results, what happens is samples with few components are used, like those which have already been purified by other chromatographic techniques, and it is actually suitable for the final polishing step in a complete purification scheme. Okay, like SSC is usually done in the end to remove salts, purify, like have a monomeric form of your protein in the end, and then use for storage or for further assays. Okay, this is the same thing that we looked at earlier. You're injecting your sample. Okay, and this is one column volume. That means this is the total volume of the packed resin, SSC resin. So the higher molecular weight or samples having higher hydrodynamic radius will elute faster because they don't have the access to the resin pores, right? So it will come out of the column much earlier as compared to the like lowest sized molecules, which have the access to pores and they pass through the pores and eventually elute out at the end. Okay. Now another is analytical SEC, or what happens here is it's a high resolution size based separation where we aren't really, usually we do not fraction it. And analysis is often connected to highly selective detectors, for example, mass spectrophotometers and photodiode arrays, right? And also there is something called multi-angle light scattering or MALS, or there are fluorescent detectors as well. Okay. This is usually performed or usually done to check the sample quality or to study the properties of the biomolecule of your interest. Okay. And again, this also requires like really small sample volumes, even smaller than the preparative one, okay, which is 0.3% to 0.5% of the total column volume, the volume of the packed SEC resin, and the column and in the sample should be passed uh, at low flow rates using long columns, and particle size is commonly in the range 4 to 12 micrometers and provides high resolution, and it can also be used for rapid purity checks and screening. Shorter columns of typically 15 centimeters provide adequate resolution, and short cycle times also saves a lot of time as well. With small sample volume requiring really small sample volume and small volume of buffers as well. Okay. For quick sample check and for quick screening and purity checks, we can do analytical efficiency. And shorter columns typically are used for purity check or screening, but it gives actually lower resolution than longer columns because in longer columns there are higher number of plates. It has more time to really resolve better. The particles inside the complete sample, particles of different sizes, have enough time to really resolve. Okay. But eventually another disadvantage of having like longer columns, if your sample is kind of diluted. So it also happens that if the column is long enough, your sample actually dilutes out, and what eventually you get out, what dilutes out of the SSC column eventually is having a quite low concentration of a molecule of interest. So if the injected sample has already a low concentration of your sample of interest, or suppose your protein of interest, and then you are running really longer columns, and then eventually it will dilute out even more. Okay, that is also like that can also happen. So it basically you need to optimize the purification step. Okay. Then there's something called desalting and buffer exchange. So whenever the buffers that we are making, it should have an appropriate composition of salts as well. Not only maintaining the pH is important, but along with it, we have to maintain the salt concentration as well. Okay. But however, the constituent salts of the buffer might not really be suitable or optimum for the downstream assays that you might want to perform with your protein of interest, right? So those salts have to be removed eventually. Okay. So what happens there is something called desalting. So desalting can also be done using SEC columns. Okay. There are columns for desalting specifically. And then also the storage. For suppose you have purified a protein. Now you want to store that protein. Now, usually what is done is you have done SSC, you have eluted out your protein of interest, like you have got really good pure protein, good quality of protein has eluted out. Now what happens here is the buffer that was passed through the SSC column to elute out your protein of interest isn't really optimum for storage for longer periods of time. Okay. So usually proteins have to be, because proteins are not very stable for really long durations of time. So what usually people do is if they purify large amounts of protein and then those are aliquoted and stored, flash frozen rather, and stored at -80 degrees Celsius for longer shelf life. Okay. Now for doing that, like once you take out the vial and thaw the proteins, thaw your sample, so you need to have a cryoprotectant, right? So usually glycerol is added to um, as a cryoprotectant so that your sample doesn't get hampered upon freeze thaw cycles during freeze thaw cycles, right? So during that, but while running your buffer normally during purification for elution, for equilibration, if glycerol is there, glycerol is a very dense liquid, right? So what happens is it can actually clog the pores of your chromatographic resin or chromatographic matrix. So what happens during that time is that's why there is something called buffer exchange. So once your final protein, you have the final purified protein in your elution buffer, then you have to exchange the buffer with a with the buffer that you will eventually be using for storing your molecule of interest. Okay, suppose your protein you're purifying, and then so it should ideally be having a cryoprotectant for freezing the sample, right? So for that, the buffer that was eluted out, glycerol is added in appropriate amounts, usually 5 to 10% of glycerol is added, and then it's buffer exchange so that your final protein, your purified protein, is in a cryoprotective environment. Okay, so um, large sample volumes up to 30% of the total column volume can be applied at high flow rates using broad, short columns, and particle size is relatively large during desalting and buffer exchange, providing low column back pressure and high flow rates. So for SEC, we saw that we have to keep the flow rates uh, like little low. But for desalting, there are columns for desalting, which removes the salts. Okay. So definitely for even when you load your sample, as compared to the size of a molecule of salt and a molecule of protein, which is nicely folded and all, it's way huge than the dimensions of a molecule of salt. Okay, or in general, so the salts will. So what will happen here is your protein will elute out faster, and then later any small molecules etc will elute out later. Okay, and hence there will be a nice separation. Like you will eventually get only your protein of interest in an appropriate pH, and then you can eventually buffer exchange to an appropriate pH buffer and store it for longer time periods. Now, so this is what I was talking about. Suppose your protein has been eluted in a proper elution buffer having different kinds of salts. But now for storage purpose or for your downstream assays, these salts have to be removed. Basically, desalting has to be done. So when you inject your sample here, it has the mixture of both the protein and the salts. Now, because the protein being really large in size as compared to the salts that constitute the buffer, the protein will elute out much faster, much earlier as compared to the salt, and hence it will be separated out. Okay. So larger molecules have no or limited access to the resin pores, which I was talking about earlier, and hence it is eluted at just after the void volume V naught. Okay, void volume means whatever is outside the packed volume of the pores. Okay, so it doesn't get to enter the pores, it passes out of the pores, passes out of the spaces outside the pores. Okay, the molecules which are really large enough and cannot enter the matrix pores or resin pores. Okay, so these large molecules are eluted out much earlier, and small molecules like salts have full access to the pores and move down the column more slowly, and hence take longer time to elute out of the column. So these molecules usually elute just before 1 CV of buffer. 1 CV means column volume of buffer has passed through the column. Okay. And this is the absorbance versus, column volume versus absorbance graph, which you actually see and monitor to check if your protein has eluted or not. Okay. And this is basically the same thing that is being shown here. And so molecules that are larger than the largest pores in the matrix in SEC resins cannot enter the matrix. Okay. And these molecules are eluted together in the void volume, the volumes which are outside the pore volume. Okay. V naught as they pass directly through the column, because these molecules are too large to enter the resin pores. And for a well-packed column, the void volume is equivalent to approximately 30% of the total column volume. Okay, so V naught is actually like nearly equal to, what we can write is V naught is nearly equal to 0.3 into like total column volume, or VP, right? We can mathematically represent it in that way. And molecules with partial access to the pores of the matrix are separated and eluted from the column in order of their decreasing size, right? So small molecules like salts, which we looked at earlier in the chromatogram, small molecules like salts have full access to the pores and are not separated when we move down the column, right? And these molecules usually elute at the total liquid volume or slightly before 1 CV of buffer has passed through the column, right? So this is what I was talking about. If this is the complete matrix, this is the complete resin, chromatographic resin, what we are talking about, and these are the pores of the resin, right? So whatever volume is outside these packed pores, okay, this is called void volume of V naught. And total bed volume definitely will include both the extra pore, the like volume outside the pores, as well as the pore volume, and hence the pore volume will be the total bed volume VC or VT minus the matrix that is there and minus the void volume. Okay, this is what will the pore volume will be. Okay, and the elution volume actually varies with the total volume of the packed bed, right? And the way in which the column has been packed, right? Hence, the elution of the sample is actually it's a good idea to always denote it using distribution coefficient or KD. So distribution coefficient or partition coefficient is actually independent of the column dimensions, and hence it helps in comparing and predicting the columns and your purification profile of your biomolecule of interest with different sizes if the same resin is used. Okay. So distribution coefficient or partition coefficient uh, that of a molecule which is distributed between the two phases, mainly the mobile phase and the stationary phase or the matrix, is denoted as concentration of the solute in the stationary phase, the matrix, upon concentration of the solute in the mobile phase. Okay. And the stationary phase of the matrix or the resin that we are talking about actually corresponds to the pore volume, right? Only this one, this one. Okay. Not the void space, not the void volume. We are not talking about the void volume here. Okay. So the stationary phase corresponds to the pore volume, that is the volume of solution inside the particles or the pores that make up your chromatographic resin. And molecules with access to the entire pore volume elute at Vt. Okay, these molecules can distribute themselves freely between the mobile and stationary phases, right? Because they have full access to the pores. Their dimensions are comparable to the pore size or much smaller than the pores as well, right? So in other words, if we talk about the partition coefficient or distribution coefficient, it represents the fraction of the resin's stationary phase that is available for diffusion of a given molecule, right? Um, so if we talk about this, what happens here is VE minus the void volume divided by the divided by the pore volume. Okay. So when we talk about the pore volume can be again broken into this one here. This is pore volume. So VI is equal to VC, means the total bed volume, minus some volume will be occupied by the matrix, right? And the void volume that needs to be subtracted, right? So now what we get here is the total pore volume can also be written as total volume minus the volume of the matrix minus the void volume. Okay. So the large molecules passing directly through the resin, through the chromatographic resin without entering the particles, with particles as in the particles here that we are talking about is the part like resin particles. Okay, that is packed there. Okay, without large molecules that actually pass through the resin without entering the pores will have distribution coefficient or partition coefficient equal to 0, right? Because it's not even getting partitioned as such, right? Those are just coming out, like passing outside the pores completely. They don't have access to the pores because their dimensions are too large as compared to the dimensions of the pore of the resins. So what is happening here is KD is equal to 0, but the molecules which have a comparable size or are really smaller than the pore size of the matrix have full access to the pores of the matrix or the particle in question, and that will eventually have a partition coefficient or distribution coefficient of 1. Okay. Now, when we talk about SSE resins, how should it be selected? So that the really important components that you are interested in, that you want to purify, are found in the most linear part of the selectivity curve or of the chromatogram with minimum peak broadening. Like eventually it should be separated out in sharp peaks and well resolved peaks. Like there shouldn't be overlapping peaks, so that your molecule of interest or your biomolecule of interest is mixed with some other contaminant. So the lowest molecular weight substances should be eluted near the near one column volume of buffer. When the one column volume of buffer passes, usually the smallest molecular weight substances, like suppose during desalting, what we were talking about, the salts usually elute near this particular volume. Okay. So when we talk about really ideal conditions, no molecules should be eluted with a distribution coefficient greater than 1 or less than 0. And the steeper the selectivity curve, the higher the resolution, we can actually achieve a higher resolution and higher separation or well resolved peaks, and well separated, well resolved and narrower peaks. Okay. Now, in cases when the distribution coefficient is actually greater than 1, what is happening here is many times, uh, because practically most of the times when we really perform the experiments, we actually perform gel filtration or SEC, situations are far from ideal. Okay. Theoretically, uh, many things can go haywire, and what also happens, that also happens in case of distribution coefficient. Many times distribution coefficient is found to be greater than 1. So in that scenario, what happens is sometimes the sample that has been loaded and the molecules that constitute your sample can show non-specific adsorption onto the chromatographic resin. Okay. In those scenarios, in those conditions, the partition coefficient usually gives a value of more than 1. And in cases when the coefficient is actually less than 0, what happens here is channeling in the chromatographic bed might have occurred and the column must be repacked. So other than the void volume, there are channels inside the, like it might have broken, the complete resin might have, detached, or the packing has been loosened. So there might be channeling inside the resin, chromatographic resin, which can lead to a partition coefficient of less than 0. Okay. So in a very ideal condition, uh, while performing SSC, only steric effects contribute to the separation of biomolecules, mainly the hydrodynamic radii of your biomolecules constituting the sample is taken into account while separating. And separation generally is insensitive to the eluent composition, right? The buffer that is passing through during equilibration or the running buffer composition generally should not affect your uh, SSC separation. And this is an ideal case scenario, but in practical situations, during non-ideal behavior, there might be interactions between the resin and the biomolecules of the sample that has been loaded, and hence the separation of your sample of interest or your biomolecule of interest actually is affected. So it is usually uh, something to be concerned about while separating highly charged substances at an ionic strength that is actually too low. Ionic strength, as in the running buffer, you have um, actually included salts of really less concentrations, or when separating aromatic molecules on dense resins with high matrix content. Okay. So also there might be hydrophobic interactions in some cases, which can usually avoided by changing. All these things can be actually avoided or prevented by changing the running buffer. And small biomolecules are more susceptible to interactions with SEC matrix or SEC resins as compared to the larger ones. And definitely the molecular shape can also sometimes explain an unexpected elution position, right? What is the folded, like, what is the folding status of your protein of interest that you wish to elute out? It might suppose be in a partially folded form. That can also affect the elution profile, FFA elution profile of the protein that you wish to purify. Okay. This is the same thing that we looked at in the earlier tutorial class, resolution, which is made how this is represented here, where the width of the peaks and the separation between the peaks are taken into account and it's averaged out here, giving us the resolution. Okay. And basically resolution represents the selectivity of the resin and how efficient is that particular matrix in separating or resolving two different biomolecules of different sizes, and how sharp or narrow the peaks is with minimal peak broadening or zone broadening. Okay. So the homogeneity of the packed bed or the packed resin and of the particles actually influence the uniformity of flow through the column, and hence it actually eventually influence the shape and width of the peaks in a chromatogram, right? And SSC, when we talk about SSC resins or SSC matrix, which are really uniform, the particles or the matrix components are actually really uniform, and there is a narrow particle distribution in while in the packed bed or packed resin, then actually the sample and the biomolecules of our interest uh, can elute out in sharp peaks. Okay, the resolution will be much better. And when we talk about SSC resins with smaller particle sizes or smaller uh, particles which actually constitute the SSC resins in question, it facilitates the diffusion of sample molecules in and out of the particles much more frequently, right? And hence reduces the time to achieve equilibrium between the mobile and the stationary phases. And so the peak width is reduced and resolution is improved. And sample dilution is inevitable because diffusion is occurring. Okay. And to minimize sample dilution, what also can be done is you inject like the initial injection volume can be used, like a higher injection volume can be used, but definitely within the limits that is like optimum for the SSC reason in question. Okay. And the resolution required between the peaks of interest should be kept in mind while injecting your sample. Okay. Usually you can have the maximum amount of sample injected. However, one should be careful of the resolution limits of the particular packed resin that we are talking about. Okay. And usually another term that people use in general, like which is also used in literature as well, it is something called as a zone. Like rather than sample passing through the SSC matrix or SSC resin or in general any chromatographic resin, it can also be regarded as a zone passing down the column. Now, for sample and buffer preparation, removal of particles in the sample in the SSC running buffers and cleaning solutions is actually really important, and because these particles can eventually clog the pores that are there in the matrix, and the matrix pores or SSC resin pores are what is the basis for separation in SSC chromatography, right? So that is, so removal of some particles in the samples, buffers and cleaning solutions, which mainly anything that has to be passed through the SSC column should be removed of stray particles or contaminants, and it should be clarified as much as possible before loading it onto the sample or before passing it through the sample, through the SSC resin. It will actually minimize the risk of blockage and also reduce the need for stringent washing procedures. I was talking about like really high concentrations of salt solution passing to be passed through the chromatographic columns after a protein prep or any other prep of your biomolecule of interest. So eventually, the more stringent washing procedures we are using, the user is using, it will eventually harm the resin or the matrix of the chromatographic column. So if we minimize that, eventually the lifetime or the shelf life of the column matrix will be extended. So what one does, especially before SEC, is using high quality water and chemicals, and any kind of solution that is to be passed through an SSE resin or SSE matrix should be filtered through filter papers or filter membranes. I was talking about like filter membranes of different sizes before use, and then air bubbles should also be removed, which is called degassing. So these buffers have to be degassed before any SSE separation, because air bubbles can also reduce the performance and separation efficiency of the SSE resin. And so eventually what also can be done is degassing and filtering can be clubbed together if filtering is done under vacuum. Okay. And buffers and columns should have the same temperature before use. Definitely, this should be kept in mind. And rapid changes in temperature can affect the packing of the resin, SSE resin especially. For example, removing packed columns from a cold room and applying buffer at room temperature can uh, have a possibility of introducing air bubbles, okay, in the packed matrix, packed SSE resin. Hence, eventually it will again result in poor separation. And samples, that's what we were talking about, clarifying a sample. Samples must be clear and free from any particulate matter. So even before, so all these filtering buffers, degassing buffers, we talked about clarifying the buffers and any kind of cleaning solutions. But in case of samples also, many times proteins can form aggregates, etc. Right? So all these can eventually affect the, can lead to blockage and affect the separation by the SSE resin. So what also happens is, before applying the sample, it is centrifuged at high speeds to pellet down any kind of larger aggregates that might have formed, and the clear solution is injected. Okay, clear sample is injected into the SSE resin for gel filtration. Okay, and yeah, this is what we were talking about. Filtration actually is removing particulate matter. Many times there are syringe filters which are actually used at the tip of the syringes after removing the needle, which gives the least amount of non-specific binding of proteins. Definitely during filtration, whenever we are using any filter membrane, so all these membranes should be made up of such materials or such substances which show like really less or like nil or zero interaction with your biomolecule of interest. So least amount of non-specific binding uh, is actually shown by cellulose acetate, regenerated cellulose, or PVDF, okay, polyvinylidene difluoride. So for sample prep before chromatography, a particular suitable pore size, filter pore size should be chosen, uh, keeping in mind the bead size of the chromatographic medium. Okay, what is the packing, like packed pore diameters or matrix pores? What is the dimension that has to be kept in mind while filtering your sample? Okay. Now, factors that actually influence the resolution during chromatography. Resin related factor, definitely. What is the selectivity? What is the match between pore size and what is the size of the target molecule? There should be a compatibility. Particle size, definitely, and particle uniformity. And column related factors also affect how long is the column, mainly how, like what is the bed height, and how well the column is packed, how well the resin is packed. That also determines or influences the separation or resolution. And chromatography system related factors, like there are tubings all around, okay, in a chromatographic system. So how wide or how narrow the tubings are, that also affect how smoothly your sample passes through, or how like that also actually regulates the flow rate as well of your sample. Many times you might not have a proper system to regulate the flow, might not have a pump. You usually use like the natural gravitational force to let the and Bernoulli principle to let the solution pass through your chromatographic resins, to especially during those times, the tubing dimensions, the diameter and length of the tubes through which your column is connected and things are eluted out, that plays a significant role. Okay. And experimental related factors, definitely the flow rate, how much sample you are applying, the flow rate. Suppose when you are passing your sample at a very, like loading your sample and the flow rate is really high. So during that time, one thing is there will be high pressure across the column matrix, definitely. And another thing is your sample is not getting enough time to really interact with the resin, especially in cases where the molecule of your interest actually has to interact with the resin or the matrix. If the flow rate is really high, what will happen is your sample of interest, or suppose your protein of interest, will not have enough time to really interact with the matrix. And the pressure through which, the force through which, because of the high flow rate, it will eventually be eluted out rather than getting the chance to interact with the matrix and form those transient interactions. Okay. So one thing is that sample volume we talked about, viscosity is talking about, right, during buffer exchange. So if one usually adds glycerol, like glycerol at concentrations that is used for storage, during those times the viscosity actually increases. Eventually, it will block the matrix pores. Okay. So the viscosity has to be kept in mind, the buffer composition has to be kept in mind. All these things eventually will affect the resolution, how well the matrix or chromatographic resin is able to separate out different biomolecules. Okay. So a critical variable is the viscosity of the sample, which are related to the running buffer. So what here we can see, if there is low viscosity, less viscosity, the peaks are well resolved here, right? And intermediate viscosity, there is some kind of band broadening here or zone broadening here. And when highly viscous buffers are passed through, okay, what happens is the separation, the resolution decreases a lot, degrades a lot, and resolution drops down by a huge amount. Okay. So elution profiles, this is basically the blue one shows the elution profile of hemoglobin and the red one shows the elution profile of sodium chloride. Hemoglobin being a larger molecule definitely will elute out faster as compared to the smaller salt, sodium chloride, which will elute out much later. However, under conditions when the buffer has like really high viscosity, what will happen is the elution profiles eventually merge. There will be overlapping peaks. The resolution is too low and too bad, and hence you won't be able to get a pure desalted hemoglobin here. Okay. Now, there are different kinds of SEC resins. Okay. Mainly these resins are made up of cross-linked agarose, cross-linked dextran. Okay. All these are there. And so based on the, even based on the dimensions, like what is the height, what is the diameter of the column, there are different columns that come. Now, how do you really know that? Suppose now a mixture of all proteins are passed through a particular column A, which is here. Suppose this is the column Superdex 200 that we are talking about. Now mixture of biomolecules are loaded and passed through this SEC column. Now you see here the numbering 1, 2, 3, 4, 5, 6. Okay. So what is happening here is 1 belongs to this particular molecule, okay, which has a molecular weight of 970 kilodalton. Okay. And 6, definitely sixth one will have the lowest molecular weight and it will elute out at the end having 1.3 kilodalton molecular weight, okay, which corresponds to vitamin B12 in this case. Now what is happening is, how will you know? Now you don't know how will the SSC elution profile of your protein of interest or your biomolecule of interest will look like. Okay. So this is usually always given with the protocol or manual of the particular SEC column. So each column has different resolution efficiency and separation efficiency. Okay. So for this particular SEC column, What is happening here is this is how the peaks will look like. So all these individual peaks belong to a certain molecular weight um, of biomolecule or protein. Okay. Here all these are proteins that has been shown here. So now you wish like your protein of interest that you are purifying belongs to like has a molecular weight of around 20 kilodalton suppose. So you can eventually understand if so your 20 kilodalton protein should lie in this range, okay, between 4 and 5, which is 66 kilodalton, and this fifth peak corresponds to a protein of 17 kilodalton. Now your protein is 20 kilodalton, so it should ideally elute out before the 17 kilodalton molecule, right? So it should eventually your protein of interest, which is 20 kilodalton molecule, which has a 20 kilodalton molecular weight, should have a peak somewhere just before this fifth peak. Okay, so this is how you will look at where exactly, and this is the elution volume. Okay, so it should elute out at this particular range, like 15 ml to suppose 17 ml or 10 ml to 15 ml. It should elute out in that particular range, a molecule of 20 kilodalton weight. Okay, so similarly, this is another super 6 increase SSC resin. It has a completely different elution and resolution profile, and based on what is your molecule of interest or biomolecule of interest that you wish to purify and what is its molecular weight, based on this profile you can eventually find out what will be the elution volume of your protein of interest. Okay, now Supertex 200 increase, this one, and Supertex 6 increase are resins which are used for small scale preparative purification as well as analysis and have the same high flow agarose based matrix. Okay. Particle size, average particle size is approximately 8.6 micrometers. So, but these two differ in their fractionation range. Okay. See, both of these here 10 by 300, 10 by 300 is the diameter and the length. Okay. So even after having the same dimensions, they differ in their fractionation range, which results in very different chromatographic profiles for the same sample that is loaded here. So this is a very different chromatogram as compared to this, or even though the samples are the same, both these represent the same ones, the same molecules that are same proteins that are eluted out of uh, these two resins. It's the same set of molecules, same set of proteins. However, the elution profiles look very different. The resolution efficiency of both these resins are very different for these 6 proteins, right? So they have a different fractionation range and a very different chromatographic profiles. And Supertex 200 increase, the first one, gives excellent resolution. Actually, you see the resolution is like the peaks are really well separated for molecular weight less than 44 kilodalton. Like after 3, you see the 4th, 5th, and 6th ones are really well separated, which means this particular resin, SSC resin, is able to uh, very nicely separate the lower molecular weight compounds or lower molecular weight substances or particles as compared to the ones which are like really larger sized, like large sized ones. Okay, and so while larger biomolecules are eluting together, right? There is very bad separation. The resolution is too bad as compared to these ones, right? For 4th, 5th, and 6th ones, which have a really less molecular weight. Whereas if we talk about Superose 6 here, on the other hand, it shows good separation between the largest molecules. So the type of resin, the packing of resin also affects SEC separation. Okay. Now this is some examples. These are some examples of chromatograms that can help you identify whether the resolution or the separation, SEC separation has been good or bad, or it's satisfactory, or it has to be performed again. Like this is a satisfactory separation with really well resolved and symmetrical peaks. This is a poor resolution. These are well separated, right? But at this point, um, this is a poor resolution. There can be asymmetric peaks as well. You can see tailing as well. What happens during tailing actually is either the column is underpacked, okay, or a column is packed at a very low pressure or at a very low flow rate. So if the resin is evenly packed, we have to ensure that. And tailing peaks can also um, be due to uneven sample like due to uneven sample application, or if there is any kind of interaction of the sample of your sample of interest with the column. Okay. So there is, in case of asymmetric peaks, there's tailing. There can be fronting as well. Okay, opposite of tailing on the other side, you can see an extended fronting, which can again caused by an overpacked column. Okay, that is a column which has been packed at excessive pressure or flow rate. Okay, also many times the peaks appearing before the void volume indicate channeling in the packed bed, right? And there can be late elutions as well. Peaks which are eluting after 1 cb of buffer can be caused by, because definitely, which means if it is eluting later, so some kind of interaction might be going on with the sample or biomolecule of interest and the SEC matrix through which your sample is passing through. Okay, so peaks which are eluting after, like showing late elution, can be due to any kind of non-specific interactions with the chromatographic resin. So usually one includes a wash step or extends the elution to ensure elution of late eluting molecules. Also, sometimes playing around with the buffer composition can help. Okay. It again depends what is the molecule you wish to purify, what is it that you want to purify, and what do you want to use it for in the downstream, what kind of downstream assays you want to use it in. Fine. Now there can be different kinds of applications for gel filtration chromatography. Uh, you can determine the molecular weight in the same way that I showed earlier, right? Based on uh, the time or the elution volume at which your molecule of interest is eluting out. And the molecular weight and size of a protein definitely is related to the shape of the molecule. And this is actually having there is a relationship between the molecular weight and the radius of gyration, and which is where the radius of gyration actually is directly proportional to the molecular weight raised to some constant, raised to the power of constant here. And this particular constant actually depends on the shape of the molecule. Okay, and the constant varies for a rod shaped molecule, a coiled uh, like for spherical molecules, for a coil molecules, and etc. Like different kinds of shapes can have different kinds of constant here. Okay. You can also determine the oligomeric status of the protein via SSE. Okay. So the native molecular weight, definitely, if your protein is actually is a monomer, is eluting out as a monomer, and if it is making, if it's dimerizing or making higher order oligomers, what will happen is eventually the overall hydrodynamic radius, overall size of the complete complex, like an oligomer or trimer or tetramer or trimer, all these things will have eventually a larger hydrodynamic radius, larger size, and hence it will elute out much earlier as compared to the monomeric form of your protein. Okay, if we are talking about protein, you are interested in purifying the protein. So in that scenario, you can eventually have multiple peaks if the protein is showing a tendency to oligomerize. Okay. So native molecular weight determination by gel filtration, uh, one can also guess the molecular weight depending on when the protein or when the molecule is eluting out, what is the elution volume. And also, we looked at SDS page in great detail. So at what uh, point of time, like what is you can determine the molecular weight at which your eluted biomolecule is running. Okay. So that can be done. And oligomeric status definitely can be checked using this formula, where you are getting, suppose, a single peak at this. This is the molecule you are getting eluting out at this particular volume or at this particular time. So it is having a corresponding molecular weight, right? But upon running on SDS page, what happens is SDS page is a denaturing page, like denaturing electrophoresis type. So what is happening if there are oligomers that are being formed? It actually, or your protein is dimerizing or something, then it actually ruptures the native state of the protein, and your monomeric form of protein will run on STS page. So that will give us the monomeric status of the protein that you are purifying, and the gel filtration will give, in case it has a tendency to oligomerize, will give the molecular weight corresponding to the oligomerized protein, right? So if we divide it, then we will get the oligomeric status of your protein of interest. Another thing that usually people do is to study protein folding. Now, suppose if your protein of interest is incubated at different concentrations of urea. Urea is a denaturing agent, okay? It denatures protein and ruptures protein folding, okay? So, if at different concentrations of urea for a particular amount of time, for a particular period of time at a particular temperature, you are treating your sample and passing it at different time points through the SSC column. Fine. Now, uh, that SSC column, the profile of that SSC column, SSC, will actually help you understand the protein folding status, like under the presence of different concentrations of your denaturing agent, right? So the gel filtration column should be equilibrated with the buffer containing urea, like the same buffer in which your sample is in. And as the concentration of the denaturing agent, which is urea in this case, is increasing, the protein will eventually unfold, and hence the overall hydrodynamic radius will increase, right? So the increase in hydrodynamic surface area will eventually lead to uh, like earlier elution, because if the protein is getting unfolded eventually, so it will have less access to the resin pores. Okay, so as a result, the protein peak keeps on shifting towards the left. Fine. So at the highest concentration of urea, the protein will have a maximum, will show maximum unfolding, right? And hence, or either maximum unfolding or completely, it will be completely unfolded, and hence most of the unfolded protein at the highest concentration of the denaturing agent will elute out in the void volume, right? It won't be able to enter the pores at all. Okay. You can also check the protein ligand binding using gel filtration. So again, if the concentration of if the concentration of the ligand is increased and the protein actually shows like it binds to that particular ligand. So overall, if the protein ligand moiety will have a larger hydrodynamic surface area, and hence it will elute out much later as compared to only protein and only ligand. Okay. If the ligand is binding to the protein, it will form a larger complex and increased hydrodynamic radius, and hence the protein peak will shift towards the left side, right? It will have all these bigger complexes will have uh, lower access to the resin pores. Okay. Now next, coming to affinity chromatography. Um, So this actually works on the principle where there are mutual recognition forces between a ligand and a receptor. It can be salt bridge, hydrogen bonding, van der Waals interaction. There can be pi-pi interaction as well. Okay. So the major determinants um, are which are responsible, provide specificity, like shape complementarity. There can be electrostatic interactions, hydrogen bonding, van der Waals interaction between the ligand and the receptor pair. Okay. So definitely, if we talk about the dissociation constant here, it will be how we, or like at equilibrium, how we denote it using individual concentrations, like the product of individual concentrations of the reactants upon the concentration of the product, or mainly the ligand receptor pair. Okay. So it's the same thing as earlier, like what we look for hydrophobic interaction chromatography and ion exchange chromatography. You have to equilibrate the resin matrix. So affinity medium is equilibrated in binding buffer, and then you load your sample. Sample is applied under conditions which favor the specific binding of the tagged protein to the ligand, and unbound material is washed away. There is this washing step, and then you elute out, where you again change the buffer conditions to promote the elution of the tagged protein, where the interaction of the tagged protein with the matrix is lowered and your tagged protein is eventually eluted out. Okay. So this is the overall chromatogram, how an ideal affinity, like how an ideal chromatogram should look like after affinity purification. Um, like you are injecting the sample, you have equilibrated, you injected the sample, and eventually you wash it off to remove any unwanted or unbound material, and then you elute out your protein of interest. Okay. So there can be advantages and disadvantages as well. For tagged proteins, there can be many advantages. It is a simple purification, and in detection of the tag instead of the target protein allows for a generic detection method, and solubility and stability can also be improved using different kinds of tags, and targeting information can be incorporated into a tag, like where should the protein go. Okay. Marker for expression can also be provided using different kinds of tags, and some tags also help in strong binding to the chromatographic media or chromatographic matrix in the presence of denaturants, making on column refolding possible, right? But there can be disadvantages as well when we really want to do structural studies. Okay. So the tags, the tagged protein, the tags on a protein can interfere with the protein structure and can eventually introduce some kind of artifacts. And also tags can affect the folding, can also have effects on the folding and biological activity of the protein that we have purified. And if a tag needs to be removed, the cleavage might not always be achieved at 100%, and some amino acids might be left. Okay. So this is again based on what kind of protease you are using to cleave off or separate out the tag from the purified protein. Okay. And in case of untagged proteins, definitely the advantage is there is no need to remove any tag, but the detection and purification becomes a little tedious many times, but not always. And there can be problems with solubility and stability, which might not be uh, so easy to overcome without putting any tag. And hence, the yield of the purified protein lowers. Okay. So, overview of sample preparation. If you wish to have an intracellular expression or extracellular expression, the overall thing is for intracellular expression, something which is insoluble in the cytoplasm, utilize the cells, cell debris, or soluble in the cytoplasm, or some kind of you wish to purify a protein which is in the periplasmic space. You first have to disrupt the cell wall and then remove the cell. Okay. And then recover the clarified sample and then move forward with downstream process. This is just an overview of what all purification and clarifying steps we have been talking about, a kind of a flow chart. Okay, summarizing the different steps. Now, major parameters to consider while preparing a sample for chromatographic preparation. This is the same thing we looked at in the previous tutorial as well. Like it has to be clarified, the target protein has to be stabilized, protease inhibition, the pH condition, the ionic state, what is the reducing whether the environment buffer environment is reducing or oxidizing, any kind of stabilizing additives, what are the conditions for purification to work, and what is the available equipment, and what is the sample size, filtration or centrifugation equipment. All these things have to be considered while performing any kind of chromatography. Centrifugation and filtration are actually standard laboratory techniques for clarifying your sample, okay, before applying onto or before loading onto a chromatographic matrix. And it is used routinely while handling small samples. Okay, uh, keeping samples on ice, even when purification is performed at room temperature, is something that has been found to increase the stability of the sample. Centrifugation helps to remove most of the particulate matter like cell debris. Filtration also removes particulate matter. And this is the same thing. Basically, the membrane filters should have the least amount of non-specific binding, protein binding. And for sample separation, sample prep before chromatography, the pore size should be kept in mind in relation to the bead size of the chromatographic matrix that is to be used for purification. Protein stability, biological activity needs to be retained after purification. So retaining the activity of the purified protein is actually uh, essential for while following the progress of the purification. Okay, so when you are detecting the target molecule, you should always be wary of whether it's functional or not, whether it's showing its biological activity or not. And you should be careful that it is not denaturing or degrading or precipitating out or showing any kind of non-specific absorption onto the column matrix. So all these eventually impairs the column function. Okay. Now there can be different types of affinity chromatography, like bioaffinity chromatography, where biomolecules are used as receptors on the matrix on the stationary phase, and the biological affinity phenomenon like antigen antibody interactions are exploited, or enzyme substrate extraction like interactions are exploited, like GST glutathione. Okay. There can be pseudo affinity chromatography where a non-biological molecule is actually used as a receptor on the chromatographic matrix, and this eventually helps in the separation and purification of biomolecules. And pseudo affinity chromatography has two specific examples, like dye affinity chromatography, where actually the matrix or the chromatographic region has a reactive dye attached to it, and the matrix bound dye has specific, like it is specific towards or specifically binds towards a particular enzyme. Like there is this Cibacron blue F3GA dye, which is attached or coupled to the dextran matrix, and this particular dye has a very strong affinity towards dehydrogenases. Okay. Then there's another metal affinity chromatography where the matrix has bound metals, uh, like transition metals like nickel, zinc, or iron, and or cobalt, etc. And these matrix bound metal ions form multidentate complexes with proteins containing like multiple histidine tags, and the affinity of the protein for the matrix bound metal is different, and these differences are been exploited in different kinds of metal affinity chromatography to purify the protein. Now there is something where thiol proteins can be purified. Okay, this is a different types of this is a very different type of chromatography where the analyte is binding to the matrix, but it is not reversible. Okay, because this type of chromatography involves the formation of a covalent bond between the functional group which is present on the matrix and the analyte that is passed through. Okay, so what is happening here is there is thiol group like SH group present on the neighboring residues, like present in the protein basically, and it is forming a disulfide bond after oxidation. So there's activated thiol on the matrix. The matrix is is activated thiol gel, and the protein is a thiol protein, okay, having SH groups. Now, after oxidation, there is disulfide bond formation under reducing environment, and the matrix in the covalent chromatography has immobilized thiol group, which forms covalent linkage with the free thiol group containing protein present in the mixture. And when there's a washing step to remove the non-specifically bound protein, a mobile phase containing compound with reducing thiol group is passed to elute out the bound protein. And then what happens here is the thiol group containing compound present in the mobile phase actually breaks the disulfide bond between the protein and the matrix here. This is SSP. P is the protein here. Okay. And breaks the disulfide bond between the protein and the matrix to release the protein in the mobile phase. Okay. So basically the buffer that is passed through plays a huge role here. Now, this is how even antibodies can be purified. Like there are beads there where antigens specific to the antibody that needs to be purified is coupled, and then thereby forming antigen cephalose, like antigen coupled cephalose beads. Then you prepare a sample containing antibodies, apply it onto the beads, wash off the unbound material, and then elute out your antigen and then regenerate the column or regenerate the Zephyrus beads. There's GST based purification where glutathione is transferase, GST, which is an enzyme. It utilizes glutathione as a substrate to catalyze the conjugation reaction, okay, for xenobiotic detoxification purposes. Now, what happens here is the recombinant fusion protein actually has this enzyme tagged onto it as a tag and is purified with glutathione coupled matrix. Now, GST fusion protein, the tagged protein, um, is actually fused with your protein of interest. Okay, GST is fused with your protein of interest, overexpressed, and then the sample is loaded onto the column, which is previously equilibrated with a buffer containing this much amount of salt. Now, during the wash step, any unbound material or unbound protein is washed off with the equilibration buffer, and then this fusion protein or tagged protein is eluted with a different concentration of glutathione dissolved in the equilibration buffer. Okay. So purified fusion can be treated with the thrombin to remove the GST tag from the protein of interest. So the mixture containing free GST tag and the protein can be purified using the affinity column again because the tag will bind to the matrix but the protein will come out in the unbound fraction. Similarly, there is nickel NTA affinity chromatography where there are beads, NTA agro or Zephyrus beads, which are charged with um, divalent nickel ions, thereby forming NINT agarose beads, and then you load your sample. This sample or the protein that you wish to purify has a polyhistidine tag attached to it, and then you let the polyhistidine tagged protein interact with NINTA matrix, and then wash off the unbound material, and then elute out with a solution which has higher affinity with the NINTA, like imidazole. Higher concentrations of imidazole are used, which removes out the bound protein and your protein is eluted out. The histidine tagged protein is eluted out. Okay. And then now what also happens is many times the receptor that or the ligand that is coupled with the matrix has like eventually the matrix get stripped off of the ions, etc., right? So the column has to be regenerated. So in cases for affinity chromatography or like NINT chromatography, what happens here is once the column or matrix gets stripped off of bound or coupled ions, NINTA coupled onto it, what happens here is it has to be regenerated. And for regeneration to occur, you have to strip the matrix completely off of the nickel ions using EDTA because EDTA is a chelating agent. It will completely chelate out the Ni2 plus ions. And then you clean it completely with a strong alkali, NaOH, and then again wash with water. And then you further recharge it or regenerate it using nickel sulfate. Like you have abundant amount of nickel ions here, and then you store it in ethanol or you can start purification or purifying it using your buffer of interest. Okay. Now, this is the same thing like for affinity column. It can be used as a tool to study or isolate interacting partner of a particular protein where the matrix is or the resin or chromatographic resin is incubated with the purified protein and then washed to ensure tighter binding. And all other sites on the bead is actually blocked with a non-specific protein like BSA or an unrelated cell lysate. So now the cell lysate or the pure protein 2, another protein is passed to the protein 1 containing beads, and then it is again followed by washing with the buffer to remove any unbound particles or unbound proteins. Now the protein 1 that is bound to the beads is eluted from the matrix either by adding a higher concentration of the ligand or under a denaturing condition. And then this eluted protein can be analyzed on the SDS page or it can be analyzed using antibodies like on a Western blot. And as a control, many times the cell lysate or the other protein that was used or that was added to the matrix in the absence of the other protein can be used to rule out the possibility of the second protein binding directly to the matrix. Okay. So another thing, what we were talking about is removal of the tag by a suitable protease and then collecting the released protein of your interest and sequencing it to see what is the percentage abundance. Okay. So now let's look at some of the sample questions. So protein was eluted at 15 ml while passing through a gel filtration column or SSC column, which has a void volume of 5 ml. Void volume means completely outside the pores, the volume occupied uh, by the column minus the pore volume and a pore volume of 1 ml. Now, what will be the distribution coefficient or partition coefficient of the protein? Okay. So the answer will be 10 here. Okay. Like eluted at 15 ml, void volume is 5 ml, right? So elution volume, void volume, you have to uh, remove it and the pore volume, which will eventually give you the distribution coefficient or the partition coefficient. Now, when we talk about radius of gyration, we looked at during SSC chromatography, the radius of gyration is directly proportional to the molecular weight, right? Where m is proportional, like m is raised to the power of a constant, which depends on the size of the molecule that is passing through. So the value of the constant is different for a rod-shaped molecule, for a coiled coil molecule, for a spherical molecule, it's different. So for a spherical molecule, the value is 0.33. Okay, this is the same from the slide previously. Now, when we talk about the reversible binding of a protein with its ligand, so which of the following will not be included when it is a reversible binding? So the answer, what we looked at the thiol binding, activated thiol gel and a protein containing an SH group, a covalent bond is formed, right, during purification. But that is irreversible. But other than that, salt bridges, hydrogen bonds, van der Waals interaction, all these kinds of interactions are actually reversible. So the correct option will be option D, covalent bond. Now a protein was run under native conditions in a gel filtration column and was calculated to be of 120 kDa in size. But the molecular weight of the same protein in SDS page was found to be 30 kDa. So what is the oligomeric status of the protein? So SDS page is a denaturing electrophoresis type where 30 kDa molecular weight of the protein is there. But however, in native conditions, the protein is oligomerizing to form a 120 kDa moiety. Okay. So the oligomeric status of the protein will be 120 by 30, which is a tetrameric form, or it has 4 subunits attaching to form a tetramer. A globular protein was incubated separately in 1 molar, 2 molar, 5 molar and 8 molar of urea. Urea is a denaturing agent for 10 hours at 37 degrees Celsius. Now, which of the protein fraction will have the least distribution coefficient or Kd value? Now, as the concentration of the denaturing agent will increase, the protein will unfold more, right? Or denature more. The protein in question or protein of your interest will denature more. Eventually, the overall hydrodynamic surface area will increase, right? So if the hydrodynamic surface area is increasing, the protein on an SSC column, the partially unfolded protein or a completely unfolded or denatured protein, uh, like completely unfolded protein will elute out much earlier as compared to a properly folded protein. Okay? So the molecules which elute out much earlier has a distribution coefficient much lower as compared to the ones which have the time to really interact and get inside or have access to the pores of the matrix. Okay, so the question being asked here is which of the protein fraction like the ones which will have the least KD value, which means it will elute out uh, like earlier or at the void volume of the resin of the column. Okay, so that will be for the highest concentration of the denaturing agent, which is in this question is 8 molar urea. Okay, because at the highest denaturing concentration, like the concentration of the denaturant, protein will be maximally unfolded and hence have the largest hydrodynamic surface area, and hence it will elute out the fastest as compared to the partially unfolded or a properly folded form. Now, in gel filtration chromatography, when elution volume is equal to the void volume, uh, like when does it really happen? So this is based on directly the formula there, the distribution coefficient there, which is the elution volume is equal to the void volume. That means the molecule that is constituting your sample doesn't even have any chance to interact or get access to the pores of the resin. Okay, so the option will be option B. Like the distribution coefficient is 0, everything is completely coming out of the void volume. Okay. Next, which of the following chromatography techniques is best suited for desalting? Okay, we have to separate out. Suppose you wish to purify a protein and you have to really have a different buffer condition for storage or for your downstream assays. You have to remove the salts. So, uh, what kind of chromatography will be good for that? It is size exclusion or gel filtration, like SSC or gel filtration. The void volume of a gel filtration column can be determined experimentally with the help of. So, this is something like a really large molecule is used called blue dextran to determine the void volume of a gel filtration column, SSC column. So what happens here is usually you first prepare the gel filtration column. Okay, you set up the column, you pack the column, set it up and equilibrate it with the appropriate running buffer, and then you inject blue dextran. You actually dissolve blue dextran in the running buffer and inject a small volume onto the column, load a small volume onto the column and start eluting at a at an optimum constant flow rate and monitor the elution profile. Okay. And blue dextran usually absorbs very highly at 620 nanometers wavelength. And you record the elution volume. Um, this volume, because blue dextran being a really large sized moiety, elutes out the elution volume of blue dextran actually corresponds to the void volume because it is too large to enter the pores of the matrix or the resin. And hence the elution volume at which the blue dextran appears is the void volume of the column. Okay, and also one few things to note is few things to note are the void volume is actually specific to the column size and what is the type of the gel matrix in question. Blue dextran actually works well because it has a really large size and it ensures it is completely excluded from the gel matrix, which basically is traveling through the spaces between the beads, not through the bead, like through the pores outside the pores. By measuring when the blue dextran is coming out of the column, one can determine the void volume of the gel filtration column. Okay. Next, stripping of nickel ions from the NTA beads is performed with a buffer containing something which will chelate out the nickel ions or divalent cations, which is EDTA. Option A. The binding buffer for NIND column should have a pH of 7 to 8. GST glutathione is an example of what kind of affinity chromatography? It's actually an enzyme substrate chromatography. GST is typed, which is an enzyme, and the substrate is glutathione. In a covalent chromatography, the matrix has immobilized dash groups. It is SS, right? It has activated thiol groups. Option B. Now, the deactivation of the CNBR activated matrix after coupling with the protein can be achieved by incubating the column with which type of substance? What is happening here is mostly this is used during antibody purification. What happens here is the CNBR mediated coupling is used where coupling is done on a polysaccharide matrix like agarose or dextran. So this CNBR is there, CNBR is added and at a pH of 11, 12 to form a reactive cyanate ester. Okay. And this further cyclizes to form cyclic imidocarbonate, and then under higher pH conditions, pH 11 or 12, like alkaline conditions, what happens is this cyanogen ester reacts with the amine group, NH2 group present, thereby forming one type of isourea derivative, right? And the amount of cyanate ester is more with agarose, whereas imidocarbonate that is formed here, the cyclic form, is more formed with dextran as a matrix. Now, what is happening here is the protein or the peptide ligand with the free amino group here the free amino group here is added to the activated matrix to couple the receptor for affinity purification. Okay. So the correct option will be option A. Fine. You need to have something with a free amine group, NH2 group, which is glycine amino acid here. Okay. Now, in the carbon mediated receptor coupling, the receptors are coupled with the matrix by a dash linkage. Now, there is this polysaccharide matrix, there's carbodiimide, and then isourea ester is formed, and some receptor having a free NH2 group is added so as to form this affinity matrix, and dialkyl urea is given out. So what happens is carboimides reacts with a matrix containing carboxyl group. Okay, this here polysaccharide matrix having free carboxyl groups to form isourea ester, and the activated matrix is then allowed to react with a receptor molecule having free NH2 or amine group, or carboxyl group can also be used, and the receptors are coupled to form to the matrix by a secondary amine linkage here. Okay. So the receptors are coupled with the matrix by a dash linkage. The answer is secondary amine linkage. Option C. Now, a protein A has a DNA binding site. So which of the following receptors can be immobilized in a matrix to be used for capturing the protein A for affinity purification? So there are, like suppose if the receptor is 5' AMP, it it actually shows affinity towards NAD plus dependent dehydrogenase. If it's avidine, it shows affinity towards biotin containing enzymes. There can be protein A or protein G, which are used for, which actually has a specificity towards immunoglobulins. There can be lysine, which has specificity towards rRNA. There can be heparin, which has specificity towards DNA binding site. So the question asks, which of the following receptors could be immobilized in a matrix to be used for capturing protein A, and like protein A for affinity purification? So answer will be option B. Protein A has a DNA binding site. So what kind of receptor can be used? Heparin. So that's it for today's tutorial session. We'll meet again the next week for the next set of chromatographic techniques. And thank you for joining. I will end the meeting for all of you. We'll meet again on next Saturday from 6. Thank you. Thank you.